Swayam Prabha. Digital India. Educated India.
In this lecture, we are going to discuss about another important spectroscopic technique, and that is circular dichroism. Circular dichroism, or in short CD, refers to the differential absorption of left and right circularly polarized light. Now, this phenomenon was first discovered by Biot, Fresnel and Cotton in the first half of the 19th century. It is exhibited in the absorption bands of optically active chiral molecules. When we say chiral molecules, it is a type of molecule that has a non-superimposable mirror images. Dichroism is a word which is derived from Greek, which means two colors, and because the sample under analysis has one color if illuminated with the right polarized light and a different color if illuminated with the left one. So, the color in fact depends on the light absorption here. Now, we see there is a growing realization of the need to perform structural studies under the conditions in which proteins are actually operating, that is they are in solution, as well as under other conditions, and to provide measures of the rates of structural changes of proteins, which are often essential to their biological function. Now, if you can recall, in previous lecture we were talking about the infrared spectroscopy, which to a certain extent helps in structural helps in providing the structural information. But circular dichroism has a lot of different parameters could be used, and lot of different conditions you can work around and get lot of useful information. Remember, these techniques are not like NMR or X-ray crystallography, which gives you a three-dimensional structure, that is exact position of atoms or mean position of atoms, but rather these are an indicative of what kind of structure a protein or other macromolecules possess, as well as it can also give various conformational changes in different conditions, which could be temperature, pH or other conditions. So, the circular dichroism has become increasingly recognized as a valuable structural technique for addressing or for these issues, which are to obtain information, structural information here. So, CD spectroscopy has a very wide range of applications in many different areas, and most importantly, like the UV CD or ultraviolet CD in ultraviolet region has been used to investigate the secondary structure of proteins. Then UV vis, we can say CD is an investigative charge transfer for charge transfer transitions and other. Near infrared CD could be used to investigate geometric and electronic structure by probing metal transitions. Vibrational circular dichroism, which uses light from the infrared energy region, is used for structural studies of small organic molecules and most recently proteins and DNA. So, there could be different variations of this CD technique as in the electromagnetic spectrum and for various applications. Now, let us start. Like we said, CD is differential absorption of right and left circularly polarized light. Let us talk little bit about polarized light. First thing is, what is a plane polarized light? Now, when you see light, it is like, as we have discussed, it has two like light can be separated into two forms of plane polarized light, parallel and perpendicular polarized light. And this, like when we consider here plane polarized light, the plane of electric vector is taken. Uh, so, we come to circular polarization of light, that is what we are going to discuss. Now, electromagnetic radiation is a form of energy, and it has both, like I said, electrical and magnetic characteristics, and electric and magnetic field that oscillate perpendicular to one another and to the propagation direction x. So, while linearly polarized light occurs when the electric field vector oscillates only in one direction. So, here when we are talking about plane polarized light, we talk about electric vector, and when it oscillates only in one plane, then it is a plane polarized light. Whereas, circularly polarized light will occur when the direction of the electric field vector rotates about its propagation direction, while the vector retains the constant magnitude. Now, remember here the circularly polarized light is generated by combining two plane polarized light, and they are differing in a say quarter wavelength, say plus 90 degree or minus 90 degree, like they are oriented, and when they combine, there is a particular resultant here, which is electric field vector, which is rotating about its propagation in a helical manner, and that is the you can say circularly polarized light is obtained. Just to understand here, if you see here, this is what we call the unpolarized light, that is it is vibrating. If we take electric vector here, so that vector or that is vibrations are in all different directions. So, it is not a polarized, it is called unpolarized light. Then if through certain means you can make light to or electric vector to vibrate in only one plane, then it is called plane polarized light. And then there is circularly polarized light, which could be in two directions. Here you can see this figure here, it shows the plane or linearly polarized light or plane polarized light, which is it is oscillating in only one direction. And if you see here, this is the circularly polarized light, and this is direction of propagation. So, like it I said, it could be obtained by combining two polarized light at 90 degrees. This is another representation here of linearly polarized light. Now, at a single point in space, the circularly polarized vector will trace out a circle actually over one period of the wave frequency. Hence, the name has come from there. So, what we have seen that electric vectors of linearly and circularly polarized light at one moment of time for a range of positions, we say the plot of the circularly polarized electric field vector forms a helix along the direction of propagation. So, for left circularly polarized light with propagation towards the observer, the electric vector will rotate counterclockwise for the left circularly polarized light. For right circularly polarized light from direction of propagation to like as observer is seeing, this is rotates clockwise. So, for left circularly it is counterclockwise, and for right circularly right circularly polarized light, it is clockwise as per the observer's position. So, as you can see, this is in a particular direction, and it will be said it is a circularly polarized light in propagating in a particular direction. So, this is how a circularly polarized light is produced. So, there could be left circularly polarized light and there could be right circularly polarized light. Now, there are two techniques here. One is called optical rotatory dispersion, and another one which we were talking about is circular dichroism. Now, most of the biological components are optically active and thus able to rotate the plane polarized light. Now, here ORD, that is optical rotatory dispersion method, measures the ability of optically active compounds to rotate plane polarized light as a function of the wavelength. But and the instrument which is utilized for ORD or which measures and produces ORD curves is called spectropolarimeter, which differs from polarimeter in that the later employs only one wavelength. Then there is another technique is circular dichroism, which where circularly polarized light is used, and circularly polarized light is obtained by, like I said, superimposing two plane polarized light of same wavelength and amplitudes which are polarized in two perpendicular planes, but there is a phase difference between the two, that is a quarter wavelength phase difference between them, and CD is observed when optically active matter absorbs left and right circularly polarized light differentially or slightly differently, and this absorption is quantified by a term known as molar extinction coefficient. For optically active samples have distinct molar extinction coefficient for left and right circularly polarized light. So, in a CD experiment, equal amount of left and right circularly polarized light of a selected wavelengths are alternatively radiated into chiral sample. Remember, only chiral samples, or here one thing has to be remembered, that these chirality can come also due to conformations or structural particular kinds of structure, and also chirality of certain molecules. So, one of the two polarizations is absorbed, that is either right or left more than the other one, and this wavelength dependent difference of absorptions are measured in CD, which yields the CD spectrum of the sample. So, every sample will have a characteristic spectrum. Now, due to the interactions with the molecule, the electric field vector of the light traces out an elliptical path after passing through the sample. Now, it is important that the chirality of the molecule can be conformational rather than structural. That is the, for instance, a protein molecule with a helical secondary structures can have a CD that changes with change in the conformation. And so, when you consider for recording this, there is one is you record the delta absorbance, or that is the change difference in the absorbance, which is delta A equals A L minus A R, which is A L is for left circularly polarized light, absorbance for left circularly polarized light, and R is A R is for right circular polarized light. So, this difference is recorded or measured, and this is a function of wavelength. So, for a measurement to be meaningful, the wavelength at which it is performed must be known, that is very important. So, because every time if you change wavelength, the absorption will be different. So, it is a wavelength dependent absorption. There is another term which is called molar circular dichroism, or it could be expressed in this form as Beer's law we have seen, that is delta A equals the molar extinction coefficient, which is the difference between the two left and right circularly polarized light, and concentration and path length. So, here if you say the delta E will be can be taken, which is the difference in the molar extinction coefficient, as we were discussing about molar extinction coefficient, if you could recall. This will be like here the difference between the two will be taken, that is left and right circular polarized light. So, this is the molar circular dichroism, and this intrinsic property is what is usually meant by the circular dichroism of the substance. So, since delta epsilon or molar extinction coefficient is a function of wavelength, a molar circular dichroism value must specify the wavelength at which it is valid. So, so this delta epsilon value is very important here. in many practical applications of circular dichroism, as we discuss here, the measured CD is not simply an intrinsic property of the molecule, but rather depends on the molecular conformation. And in such a case, the CD may also be a function of say temperature concentration, temperature, it may be a function of concentration, chemical environment around, like say for example, solvents and other things present in there. So, in the case of reported CD value, one must also specify these other relevant factors in order to be meaningful for these values actually. So, that is very important that when we are reporting CD values as we measure in terms of dichroism or in molar extinction coefficient or whatever terms, it is very important to specify other relevant parameters as well. Now, there is term called molar ellipticity. So, as we were discussing about delta A or absorbance, differences in the absorbance, which is usually measured for different reasons, but like most measurements are reported in degrees of ellipticity, and molar ellipticity is circular dichroism corrected for concentration. Molar circular dichroism and molar ellipticity are readily interconverted. So, here if you see in this figure, there are vectors ER plus EL and here ER minus EL. There is a angle here for so this is ER and this is EL here. So, this could be expressed in terms of elliptically polarized light, or as we were saying that this could be converted like two values, molar circular dichroism and molar ellipticity could be converted by this particular equation here. So, elliptically polarized light say is composed of unequal contribution of right and left circularly polarized light, and this relationship is derived by defining the ellipticity of the polarization in terms of this equation. Now, here ER and ER are the magnitudes of the electric field vectors of the right circularly polarized and left circularly polarized light. Now, the units of molar ellipticity they are mostly expressed in terms of degrees centimeter square per decimole, and to calculate molar ellipticity, the sample concentration, cell path length in centimeter, sample concentration in grams per liter and the molecular weight gram per mole must be known. If the sample is a protein, say the mean residual weight, that is average molecular weight of the amino acid it contains is used in place of the molecular weight, because essentially treating the protein as a solution of amino acids. Mean residue ellipticity is another term, like for methods for estimating secondary structures say in polymers like proteins and polypeptides in particular, it often requires that measured molar ellipticity spectrum be converted into a normalized value, specifically a value independent of the polymer length. So, mean residue ellipticity is used for this purpose, and it is simply the measured molar ellipticity of the molecule divided by the number of monomer units in the molecule. So, that way you can calculate mean residue ellipticity. Now, this is a typical CD spectrum of a protein for different kinds of conformations. Let us discuss them here one by one. If you see here, this is the solid lines here. This spectrum is for alpha helical conformation in a protein, and if you can see there are two negative peaks here, which are around 208 or 209 and 222 nanometer wavelengths, and there is a positive peak here which is around say somewhere around 190 or so nanometer. Then there is, so this is for alpha helix, then there is this particular which is dashed lines, long dashed lines are for beta conformation. Now, if you can see here, there is a peak here around 218 nanometer and there is a positive peak around a little less than 200 nanometer. So, for beta conformation. So, this clearly shows that beta sheets here they do not give a very clear absorptivity as compared to alpha helix. Then there is a this curve here, which is uh, for particular random structures. So, dotted lines here gives 310 or other kinds of structures. This cross dashed lines produces like 310 or other helical structures, and there is a negative peak below 200 nanometer, and so this could be another structure here, which is simple short dashed lines here, which is you can say irregular structure, which is present also, like if structure does not have any regularity, then this will be not show very significant or clear peaks in here. So, these are different, like if you have a protein molecule and if you are observing it in CD spectroscopy, then there are clearly signature patterns here available, like say for particularly for alpha helix here there is very clear peaks, negative peaks at 208 or 209 and 222 nanometers, which are present, and for others like beta sheet also not so clear, but still at 218 nanometer, and then others are there, which is for 310 or random coil structures which are present. This one here, one which is like dotted lines, is more of a beta turn type of structure. So, these are indicative of the kind of structures which are contained in a protein molecule. All right. So, these like as we have seen characteristic CD spectra for protein, and kind of transitions here which happen in this spectra, which is like at UV spectra we can say, where and it ranges from around say 160 nanometer to 240 or 250 nanometer. If you observe the kind of transitions here, there are like transitions which are pi transition, or they are negative at 195 nanometer in random coil, which are N to pi transition. N is non bonding to pi anti bonding transitions. For if you consider for beta sheet, there are pi transitions at 218 nanometer, that is negative peak as we have seen, and there is a positive peak at 196 nanometer, which has N that is non bonding to anti bonding pi transition. If we consider these transitions for alpha helix, there is pi transitions which leads to the perpendicular, which leads to the positive 192 nanometer peak, and there are pi transitions also for negative peak, which are parallel at 209 and 222 nanometer. So, these are few like so pi transitions you have like 192 and 209 nanometer, and negative at 220 nanometer is red shifted here. So, these are transitions which are present which occurs during this particular CD occurs in this during CD spectroscopy, when we expose the protein sample to in CD spectroscopy. Now, if there are aromatic residues here, aromatic residues held rigidly in space, then its environment is asymmetric and it will exhibit circular dichroism. Aromatics have they allow pi transition and that are directed in the plane of the pi bonding systems and are orthogonal to each other. Now, phenylalanine has a small extinction coefficient because of high symmetry, and it is also the least sensitive to alteration here. Tyrosine has lower symmetry than Phe and therefore has more intense absorption bands, It has absorption bands around 76 nanometer and a shoulder at 283 nanometer. So, the hydrogen bonding to the hydroxyl group leads to the redshifts of up to 4 nanometer. Then there is tryptophan, which has most intense absorption band centered around 282 nanometer, and hydrogen bonding to the NH can shift the 1 LA band, that is particular band by as much as 12 nanometer. Then there are disulfide spectra have a broad band at 250 to 300 nanometer with no vibronic structure. So, there are like these residues, which is phenylalanine, tyrosine and tryptophan, depending on their symmetry and asymmetry, can have absorptions maxima at different wavelengths because of their interactions and their properties. Now, if we consider the CD instrument, these are known as spectroperimeters. They measure the difference in absorbance between the left and right circularly polarized components, but will generally report this in terms of ellipticity in degrees. So, there are various methods by which CD effect can be measured in the spectropolarimeter, which could be modulation, in which the incident radiation is continuously switched between the L and R component. There could be direct subtraction, in which the absorbance of the two components are measured separately and then subtracted for each other, and there could be another way, which is like ellipsometric way, in which the ellipticity of the transmitted radiation is measured. In such a CD instrument, plane polarized light is split into the left and R components by passing through a modulator, and the modulator normally used consists of piezoelectric quartz crystals and a thin plate of isotropic material like suffused silica tightly coupled to the crystal. As the transmitted radiation is switched between L and R components, these are detected in turn by photo multiplier. Now, origin of the CD effect, the left and right circularly polarized components of plane polarized radiation. If we see here, as I will show you, the two where the two components have the same amplitude, and when they combine, they generate plane polarized radiation. When the components are of different magnitude, the resultant will be elliptically polarized. Also, when we consider absorption, that is a relationship between absorption and CD spectra, then what we will see is that the positive there will be absorption will be in the same way seen like as in absorption spectra, but CD spectra will be different. So, as if we see here in terms, if you see here there is one like both components, if they are of equivalent, then there is a plane polarized light, otherwise there is a elliptical or you can say like if two components they are not same or they are different magnitude, then there will be elliptically polarized, as in seen in here, they are elliptically polarized. Likewise, if you see absorption and CD spectrum, these are two different. Absorption spectrum every time shows the absorption. If you see here the first one, this is absorption, but this is left circularly polarized light in positive way, this is right circularly polarized light, and here there is no signal because this is achiral chromophore. So, here like in here right is more absorbed than left, here left is more absorbed than right, and this is achiral. So, these kinds of patterns are seen in CD as compared to the absorption spectrum, and if these components are different, then certainly elliptically polarized light is obtained. All right. So, if we see the simple CD instrument, it looks like a spectrophotometer like instruments with computer controlled, with the computer is there which contains software to control the instrument. In very simple terms, if it is ORD if you see here on your screen, if you are working with plane polarized light, then what you have is a light source, linear polarizer. There is a linearly polarized or plane polarized light which passes through the sample. Depending on the sample, there will be a positive rotation angle, or differently it will react, and finally it will be detected after passing through analyzer. But in circular dichroism, what is done is there should be a circular polarizer, or circularly polarized light has to be which will be like left and right circularly polarized light, and as we said, alternatively there could be a pattern where left and right circularly polarized light are put in, or there could be different ways. As this light passes through, there will be a preferential absorption for one of them, and that will be detected in here. This is very simple schematic of different things which are present in here. There will be power supply with a cooling box in here, monochromator, there is a sample holder, this PEM electronics here, and then finally you have computer system and detector in here. So, these are like a very simple way to like these are photomultiplier tube, which is for each reference cell as well as for sample. There is 2 PMT tubes in here, and it will be data will be processed and finally will be analyzed on the computer. So, these are very simple schematics. The ORD and CD spectra, if we compare say of a polypeptide which may be random, has a CD spectrum which is similar to that of simple amide, but will be bigger in magnitude, but the spectrum will displays a small positive and to pi transition, and that is transitions at approximately 230 nanometer, and a large single pi transitions at around 195 nanometer, like I have shown you in there. The spectrum of the alpha helix shows a large negative N to pi transitions at 222 nanometer, and pi transitions which is split into 2 transitions because of exciton coupling. These transition has a negative band at approximately 208 or 9, and a positive band at approximately 192 nanometer. So, 220 2 nanometer band is due to the negative band is due to N to pi transition, that is non-bonding to anti-bonding pi transition, whereas 209 or 208 nanometer and 192 nanometer band is due to pi transitions. The CD spectrum of the anti-parallel pleated sheet, as we have shown you, is pi transitions. However, the splitting of the transitions are different than in the case of the helix. The spectrum of anti-parallel beta structure, as we have shown, has a negative band at 280 nanometers. So, what we have shown in earlier in the spectra of protein CD spectra of proteins, the negative band mostly is for anti-parallel beta, and there will be positive band for 195 nanometer. So, if you consider ORD and CD spectra in here, then as you see here, these diagrams, they are clearly there are much clear bands in CD spectra, like for this is for alpha helix and this is for beta sheet, and this is for random coil structure. If you see that for alpha helix there are 2 peaks in here, but does not seem to be in ORD here. Likewise, there are like the peaks which are much more clear and distinct in CD spectra as compared to ORD spectra. So, CD spectra is now most of the time it is utilized for different applications, particularly structural or conformational determination of macromolecules and other molecules also. Now, if we consider certain applications of CD in biology or biological molecules, one is, as we were discussing and showed these spectra also, that is determination of proteins secondary structure, that is alpha helical and beta sheet content. So, that could be determined, or random coil content through signature spectra, and in standard conditions that could be determined, that what is the percentage content of say alpha helix and protein. Then one can determine the optical purity of biological molecules. Also analysis of the tertiary structure of proteins and conformational changes can be worked in here. Now, remember that is not like you obtain tertiary structure, but the conformational changes in the tertiary structure could be detected by CD experiments. Then comparison of the secondary and tertiary structure of say wild type and mutated proteins could be performed in here, doing far UV and near UV CD. Nucleic acid structure and changes upon say binding or melting could be monitored at a particular wavelength, like say DNA melting curves could be obtained in CD, and like we also discussed in UV vis, where at particular wavelength if you monitor, then like G plus C content and other things could be known actually. So, in general this phenomena will be examined in absorption bands of any optically active molecule. So, circular dichroism is exhibited, or this particular property which is exhibited by biological molecules because of their patterns like dextrorotatory or levorotatory components. So, therefore, this like alpha helical content of protein or the double helix content of nucleic acids have CD spectra, which is we can call signature patterns of these structures, and they could be very much utilized for knowing the structure of say many different biological macromolecules. Now, the capacity of CD to give representative structural signature makes it a very powerful tool in modern biochemistry, with applications that can be found virtually in every field of study. CD is generally considered to be more advanced, and its advantage is apparent in the data analysis. Structural elements are more clearly distinguished, since their recorded bands do not overlap extensively at particular wavelengths as they do in the ORD or optical rotatory dispersion. So, there could be different kinds of analysis which could be performed in CD experiments. There are far UV CD experiments like ultraviolet region 195 to 250 nanometer. CD spectrum of proteins can reveal important characteristics in far UV of their secondary structure, and in this case the chromophore in this area, this particular region is peptide bond. CD is a valuable tool especially for showing changes in the conformation. Like for example, if you want to study the secondary structure changes in a molecule as a function of temperature or as a function of pH or the concentration of denaturing agents like say urea or guanidinium chloride, this could be monitored by comparing the spectra in different conditions, and that can reveal very important thermodynamic information about the molecule. And lot of experiments for say enthalpy or Gibbs free energy of denaturation could be done, and like a lot of kinetic experiments could be performed in CD, which is like binding experiments or like monitoring at different conditions, environmental conditions, and secondary structure content. All these things could be performed in here. Then there is near UV spectral region, which is 250 to 350 nanometer, and here the chromophores, as we were discussing, is aromatic amino acids and disulfide bonds, and we have discussed about like phenylalanine, tyrosine and tryptophan. They absorb at different wavelengths, and then disulfide bonds can give. So, the patterns which are obtained for say phenylalanine, tyrosine or tryptophan can give an important information about the changes in conformation in tertiary structure because of certain shifts in their absorbing patterns actually. Then there is visible CD spectroscopy, is also very powerful technique to study metal protein interactions and can resolve individual DD electrons transition as separate bands. So, CD gives it will not give you a specific structural information in terms of 3 dimension, which could be done more clearly by X-ray crystallography and protein NMR or NMR technique, and both give atomic resolution data. But CD spectroscopy is very useful, it is a quick method which does not require large amounts of proteins or extensive data processing. It is a very direct, very convenient method for analyzing very quickly the structural content of a sample, and it is mostly utilized for to study protein in solutions and under various conditions to see the changes, to see the conformational changes occurring. This is like there are whole lot of applications for CD experiments, and there are a lot of variations in experimental ways which one can perform and obtain useful information. There are certain experimental limitations also of CD experiment, like CD has also been studied in carbohydrates with but with very limited success actually, because CD spectra which is for carbohydrate is like around 100 to 200 nanometer, where the corresponding CD bands of unsubstituted carbohydrates lie, and substituted carbohydrates with bands above this region which could be measured, but otherwise it is very hard to measure. So, measurement of CD is also complicated by the fact that typical aqueous buffer systems often absorb in the range. So, where structural features exhibit differential absorption of circularly polarized light. So, only certain buffers could be utilized, like phosphate buffer is very common, or say carbonate acetate buffers are. Here if you say phosphate and other buffers could be utilized. Certain buffers are totally like not like if you say phosphate buffer also, phosphate buffer or carbonate, they should be utilized in only very small amount or concentrations, otherwise at high concentrations they are also unacceptable. Tris buffer is completely avoided while performing UV CD. There are like certain buffers cannot be utilized at all, but other buffers could be utilized which are at low concentration. The usual light source in these instruments is a high pressure short arc xenon lamp, and ordinary xenon arc lamps are unsuitable for use in the low UV region. So, light from synchrotron sources also has been with a much higher flux at short wavelengths and has been used to record CD down to 160 nanometer. Recently, the CD spectrometer at the Electron Storage Ring Facility ISA at the University of Aarhus in Denmark was used to record the solid state CD spectra down to 120 nanometer. So, at the quantum mechanical level, the information content of circular dichroism and optical rotations are mostly identical. So, these are certain limitations which one could overcome by taking proper precautions here, and can have successful CD experiments to obtain useful data. So, this was about CD spectroscopy, and as we have seen, the CD spectroscopy is a very useful technique, and this is based on differential absorption of right and left circularly polarized light by a sample which is a chiral sample, like we have seen alpha helix and or protein secondary structure has a has signature spectra or signature patterns, and this could be very useful tool for determining the secondary structure content of unknown protein by you can take in database standard proteins whose crystal structures are solved could be taken, and these CD data could be collected for those standard proteins, and then they could be standardized for particular peaks in various conformational states, and then unknown spectra from unknown sample can be compared to those, and relative secondary content in terms of percentage could be calculated. So, it is a very useful technique. Many times you can also one can compare the protein conformation or monitor the protein conformation in various environmental conditions, under denaturing conditions or otherwise. So, biological macromolecules and other different kinds of molecules could be studied in here. Binding studies could be performed, kinetics at different temperatures could be performed. So, there are a whole lot of different applications of this technique, and this could be a substitute or as a quick technique. It could be utilized for secondary structure determination before one does or performs X-ray crystallography or NMR. So, this completes our section on circular dichroism, and in the next lecture we are going to discuss about NMR and X-ray crystallography. Also, we will be discussing about atomic absorption and flame emission spectroscopy, plus a little bit about mass spectrometry. Thank you.